So for the end of this afternoon, it's my pleasure to introduce Carla Bonetti Madelaire. So Carla comes from Brazil, now from the States, but from Brazil, where she did her PhD in Sao Paulo. And now she's in San Diego, the Institute of Conservation, as you can see, for the Wildlife Alliance of San Diego Zoo. And she will talk to us about vertebrates other than farm animals, but yet cousins. So maybe we will learn also for all our species from what she is doing on, well, animals we are not familiar with. Thank you, Carla.
Thank you. So I have the interesting, entertaining job to wake you up at this time and, engaging and talking about a topic that some of you might not seem interesting at first. I can guarantee you by the end of this talk, you'll be like, wow, I should be working with the wildlife. Clicking. Okay. So, I think a common topic between all of us, independent of you working with livestock or wildlife, is a constant crisis that global changes are, affecting humans, wildlife, and livestock. And I think what brings us together in this room right now is the fact that all of us are trying to find solutions for this pressing problem of a global anthropogenic derived changes in the globe. And what if I told you that we could actually figure this out by studying wildlife? Because if you think about it, all the solutions for these problems that we are facing in terms of climate change, for instance, have been figured out by other species. So in this slide here, we have, for instance, species that have what we call flexible homeostasis. So they are capable of fluctuating their body temperature, regardless of, being pathological. They are not in a pathological state. They can fluctuate body temperature and still be healthy. For us, humans and livestock, for instance, to increase your body temperature, it means that you're fighting an infection. It's a pathological state. So those guys over here, they can do that and not be sick. And the same thing for species that can have high diets with high levels of sugar and not getting diabetes, or species that live in high altitude or are diverse, they can stand low oxygen levels with no consequences for their health. So in this case, if we study the buffering response of those species that have a flexible homeostasis, we might be able to find therapeutic targets for pathological conditions in humans and livestock. Then by comparing with species that don't have that ability for fluctuating their homeostatic state, could be actually an answer for us to understand what it means to be resilient. So from my area, that's comparative physiology, this term of common garden experiments is very, very common. Everybody knows about that, but it was a surprise for me that other people from other areas never heard of this before. So what a common garden experiment entails is exposing a species to exactly the same condition. Like Ramiro told us about this morning when he put all the embryos in the same conditions. So that was our idea. So let's put all those species flexible and strict under the same conditions of oxygen, temperature, and glucose levels and see what happens. And of course, for logistical reasons and also ethical reasons, you cannot do that kind of experiment at the organismal level. So we had to take advantage of using lower levels of biological organization to approach this question. And we recently published, like last year, it came out this paper showing a lot of advantages of using ex vivo and in vitro methods to answer those kind of questions. And I'm sure for this audience, this is not new at all. So for all the types of cells that we could use, we chose fibroblasts. because they are obtained from no little sampling. So a skin biopsy can give you a lot of different cells, a lot of cells for you to work with. And we also have shown 2022 that those cells display several parallels with organismal physiology. So what our experiment entailed? So we plated all those species, like the fibroblasts of all these different species, and we kept them at 37 degrees Celsius, 8 millimolars of glucose and 20% oxygen levels. And then we exposed them for 24 hours to 32 degrees Celsius, 41. Low levels of glucose, so 2.5 and 30 millimolars, and 0.5% of oxygen concentration. Then here we did acute and chronic exposure instead of doing different levels of oxygen. Good, so that's a basic what we did. And we collected, physiological and morphological markers for cellular health. We also collected samples for doing an expression and also chromatin assessment. So for you to have an idea on how big this experiment was, it was about 1,900 experiments. So it was a lot. We did 5 individuals per species. So we'd also capture variation within species. So biological relevance and biological variation. So the first example that I'm going to show you about how the fibroblasts mimic or recapitulate some of the features that we observe at the organismal level is by comparing the production of lactate on fibroblasts exposed to 0.5 oxygen concentration for 24 hours. And we compare humans and rats that are not very good at holding their breath, intermediate diver, which is the humpback whale, and a deep diver, which is the webbed seal. We were expecting that humans and rats would produce a lot of lactate because they would be relying heavily on anaerobic metabolism. Whales, intermediary, and seals with the lowest amount of lactate production. And that's exactly what we saw. And that was very exciting. So that was one of the very first evidence that we had that fibroblasts could be good models, depending on the trait that you were observing. Another super interesting data that we had comparing restricted and flexible species is by doing an assay where we measured oxygen consumption, oxygen mitochondrial consumption using equipment called Seahorse. Maybe some of you are familiar with that. You just plate the fibroblasts and do measurements of oxygen and other traits of the electron transport chain. And in this graph here, you're going to see the changes in oxygen consumption. So it's close to 0. It means that it didn't change much 32 to 37 degrees to the control condition. And here we see that the flexible species, so the dromedary camel and the 13-line ground squirrel, which is a hibernating species, they are able to buffer that temperature change, so they don't show any changes in oxygen consumption. In the restricted homeostasis species, so the rhinos and the rats, show a significant decrease in oxygen consumption under the non-optimal temperature. And even more exciting, when you look at 41 degrees Celsius, you can see that all 3 restricted homeostasis species show either a decline or a significant increase in oxygen consumption, indicating that that feature also recapitulates their ability of buffering different body temperatures. We also evaluate cellular morphology. So in this picture, you can see fibroblasts that were painted nuclei in blue, mitochondria in red, and the cytoskeleton in green. And this is at 37 degrees Celsius, and here it's after 24 hours exposure exposed to 41 degrees Celsius. You can visually see with your eyeballs that this is different. We need, like, with a large data set that we have in the amount of species that we have. And also keeping in mind that phenotype is a continuum, it's not a binary trait. We took advantage of this software called Cell Profiler. So you input the pictures that you take under the microscope, you teach the software what is the nuclei, what is the mitochondria, what is the cytoskeleton. And by machine learning, you're capable of detecting even the slightly changes in morphological phenotype, changes that your eyeball you cannot detect. And we saw that 41 degrees Celsius exposure results in this senescent phenotype. So you see an enlargement of the nuclei and also a changing of the filiform shape that's classical for fibroblasts. And the senescence phenotype is also confirmed by a physiological assay where we measure beta-galactosis. So that's an enzyme that is produced by cells when they start entering senescent state. And here you have 15 different cell lines from 15 different individuals. So what I want to call attention in this data is that you can see there is some individual variation as well. So this is another thing that we're trying to capture as well, like why some individuals do better, there's not so much, for others, it's kind of the same. There's an increase in that, significant increase in that enzyme, they're 41 degrees Celsius. I think the message between all those different features that I show, physiological and morphological features that I have shown, is that yes, we are capable of recapitulating some of the physiological features that you observe at organismal level. It's also, variable, and that's something interesting to be explored. Now I'm going to go to the second part of our data set, which is looking at RNA expression or gene expression. This was done by Lucas Moreira, which was a postdoc in Eleanor Carson's lab at the time. Now he's employed a bioinformatician at Colossal. And when we got the RNA-seq data back, it was incredibly challenging because of the amount of species that we had and the different treatments. It was just overwhelming. We're also thinking, okay, if we just do an ANOVA or see what is significant change between the different species, what is differentially expressed, doesn't tell us the whole story. Because, maybe the two animals, they're going to be closer together in terms of gene expression just because, they're closer in evolutionary time. And what we were actually interested in capture with this data is what are the similarities between species that are flexible in terms of homeostasis? So are traits or genes that have been expression of genes that have evolved convergently in different lineages. So for that, we use a phylogenetic ANOVA to test for those changes in shift, so this expression shift based on correcting for the evolutionary distance among the species. And here are some results. So from this analysis, this ANOVA analysis, for 41 degrees Celsius, when you compare the RNA expression or gene expression 37 to 41, we had about 15 genes that were significantly differentially expressed. I'm hand-picking two of them just to show you as an example. The first one is the TBL2 that you can see in this graph that was increased expression in all 4 flexible species. So the bats and the squirrels that are hibernating species, so they decrease their body temperature, no problem. And the 2 camels. And this gene is regulating unfolded protein response and also activated under cellular stress. So that's one of the functions that was important for those species to maintain their homeostasis under this heat stress. Another interesting gene that was differentially expressed in 3 out of the 4 flexible species was UKCBRB. So this gene is a part of the electron transport chain. The mitochondria in their downregulation, as we can see here, is activated when the cell activates their damage response pathways. Well, but another interesting, another topic that we are interested because, especially in USA, there's like NASA is incredibly interested in trying to figure it out. How can we get humans into this space travel for a long distance? So they want to figure out how can make humans hibernate for those long space travel. So another interesting thing that we tried to see if was a direction and a specific response in hibernators when you decrease the temperature and what kind of response those fibroblasts show at cold. So here in this graph, you can see the patterns of gene expression that are far more similar between the two hibernating species. So we have a total of 875 genes with convergent shifts in expression. And that's the other big message in this talk that evolution comes up with solutions that are the same using different genes. So we have a lot of different genes here among this 800, right? So we can pick a couple of them with their different functions. So you can see autophagy, proteolysis, fluorescence, and so on. All of those different functions for the cells were important to keep homeostasis, and you decrease the temperature to a known optimum. That's not the only story that we're trying to tell. That's a lot of people, when I meet people after I give this talk and I meet one a few months later, they're like, well, have you published this? What's going on? And so on. We're still trying to figure it out, the story, because again, we have a crazy amount of data. We want to tell a compelling story using a majority of our data sets. And when we saw this expression for the RNA-seq data, like, oh, maybe it's not only that, not only the differential gene expression, because one of the first predictions that we had that didn't happen to be true, it was that we were expecting that would be one of the genes that would be differentially expressed was the heat shock protein. Like every time you talk about heat shocks, like, oh, experiments that you expose cells or animals to heat, you expect an increase in heat shock protein. And we didn't see that at the gene expression level. We saw at Western blots, but not at the gene expression level. So we're wondering what was happening, right? So one of the packages that Lucas used, and I made some notes here so I don't forget. So it's called RMETS. You can use that package to look at different alternative splicings for genes within your RNA-seq data. And then here you can see that this gene, which is the precursor of the heat shock protein, it didn't change their expression in the dromedary camel. And of course, in humans, it increased. And these different colors, they represent the different isoforms of this gene as well. And then when you look at the alternative splicing for this specific gene, you can see that the humans also do some alternative splicing for this gene, but the difference between the camels, between 37 and 41, is far more compelling compared to the humans. The story is not only about the gene expression, but also probably related to some other mechanisms and other levels of explanation that we're just not fully aware yet. I mentioned that we collected TAC-seq data, and I'm not even going to talk about this in this talk, simply because we don't have the people power to analyze all of this data. Lucas moved on to this a new position, and he didn't even have time to touch that data just yet. So I think the message here for this second part is to be aware of the fact that the stories that we want to tell, sometimes there are more layers than we can even imagine or get it out. Another super interesting and compelling aspect of this project when I started this was the fact that this project is also very, very integrative. So we have a mathematical modeling group working with us to try to model what are the response between genes and physiology under different stressors. So Christine Cruz from University of Minnesota, she got the data that I collected for humans, 37 degrees Celsius, and also the different treatments for glucose and temperature, all the physiological parameters that I collect, and also the gene expression data, and work on building a model that's called agent-based to understand what drives this response. So she interacts what are the genes that are differentially expressed at the different treatments, and you know, ask me details because I'm not a mathematical model person, but she was trying to figure out how those different elements interact. And then this was just accepted last week, so stay tuned for when it came out. It's super interesting model, and she called this model Genoscope. And Dominique told me there was an agency in France that used to call that Genoscope. Like, wow, I have no idea, so maybe we're stealing someone's patent here. And the final message for my talk is to give some advertising about my position and what I'm planning to do while I'm at the San Diego Zoo. So we do have, many of you might have heard of the Frozen Zoo before. So Frozen Zoo is a biobanking facility that has about 10,000 samples from 10,000 different species from different taxes as well. And thus far, we have like 50 years of story with the Frozen Zoo, and a team of technicians that are working, on the clock to biobank lines from all those species that are highly endangered. We have extinct species as well. Some of the resources here were so important to improve conservation genetic efforts. Two species were fibroblast lineages from this collection we use for cloning to store genetic diversity of some species that are highly endangered. And they recently hired me as a scientist to publish a research program that leverages cell culture genomics to advance conservation. So what I'm going to do is continue to understand what are the resilience mechanisms between interspecific and also interspecific. Also work on understanding what it means to have a viable cell line and genomic integrity for focusing on biobanking efforts. I'm sure that a lot of people here in this room that work with cell lines, it's like, what is a healthy cell line? It's just look with your eyeballs and tell, oh, this looks good. This does not look good. Okay, so how can we make these traits more quantitative rather than just qualitative? And also integrating features of cellular biology and genomics for species rescue and also strategies for conservation. None of this work would be possible without amazing collaborators. So the whole flexible homeostasis project was envisioned and created by Eleanor, Allison, Diane, and Valerie, amazing women that I have the privilege to be working with. And at the time when I started on this project, I was a postdoc in Allison in the lab, and the other two postdocs involved in this project as well. And if any of those aspects of this project, it's appealing to you, I would recommend for you to email these people, including myself and so on, for us to have a chance to keep thinking about these questions and how can we improve and increase those efforts in understanding resilience in different species. And this is some of the people that are collaborating within my team as well at the San Diego Zoo. And thank you so much for the attention. Please, connect with me via LinkedIn or send me an email. Let's keep in touch. Thank you.
Thank you very much, Carla. Are there questions? Yes.
You very much for your talk, Carla. I was wondering, you have so many different species, and well, it's supposed to be the same cell type. Do you? I mean, I've seen you have different conditions, but is it always, I don't know, DMEM or whatever? So the medium is always the same. You can't think that maybe some would need other requirements.
Oh, so that was one of the first thoughts when I started this project, right? Because you have different cell lines and they use different medias, right? Some of them use EME, some others alpha, others it's a mixture of everything. But we wanted to make the conditions as consistent as possible. So what we did was we bought this DMM without the sugar. And we manually add the sugar to maintain the 8 millimolar. And then we kept all the cells for at least 2 weeks, those conditions for this media. And then we perform the experiments afterwards. We would not capture, like that, this species grow better under alpha and then you change media, and then the things they're seeing is because of the media, not because of
and
the
you say
condition.
cell lines, it means that you mortalize them with a
No,
SVT?
sorry, no,
Okay,
they are
this is
primary.
just primary.
Primary,
Okay.
yes.
Thank you.
Hi, Carla, thanks.
Uh-huh.
Do you have genetic data? So do you have whole genome sequence of itself?
Yes, we did a whole genomic sequencing for each one of the 5 individuals that we have. So, it looks to be better, like why we have to do that, but we did. Yes.
Okay, because
Yes.
then there are some approaches in which you try to find if there are regions of the genome that are, let's say, accelerated to one species to another, and there are some softwares that maybe could be interesting to check.
Yeah, that's an interesting point. Thank you.
It's
Oh, I'm feeling so happy everyone's asking questions. You're so kind.
Yeah, thanks, Carla. Nice talk. My question is whether you have or will do some gain of function of your cells. Like if you found some genes that are interesting, you those genes in other species and see whether that would confer some resilience.
Yeah, that's one of the things that we definitely want to do. It's on the horizon, but it's really hard. Like for me, at least, I'm not do that kind of work, but it's one of the reasons also wanting to come here and see what people are doing. What kind of genes should I choose? All the genes that one, which one should I choose? So that's a big question, right? Does anyone have an answer for that? Because I don't.
Thank you. Attends, je redescends. Don't fight.
Thank you a lot, Carla. It was really great. I don't have really a question, but fascinated by what you said at the beginning, that actually we learn from nature, from species, how we adapted. And so from there, you got to really find the things that we want change to improve. I mean, that's fundamental. So I wonder if from this work that you are doing, we could come up with, the ideal pathways, functional pathways that should be investigated in the ideal system. I mean, of course, if you move to more complex in vitro systems than like primary fibroblast, you will get more information, more detailed things, but if in fibroblasts, you can see already all this, is that it's really powerful thing.
Yeah. One of the reasons why I have chose to show the function related to the genes that we saw differentially expressed is because of that. And maybe we're trying to find the solutions based on like thinking about the functional genomics and maybe doing this genetic engineering. But can we focus on function, and can we focus on improving these types of functional within the different species, right? So if you do improve metabolism, independent of how you do that, maybe with supplement, maybe with genetic engineer, and study that then we can understand why those pathways are so important for maintenance of homeostasis and resilience, right? Yeah, you're so right.
And I think the non-coding region will be.
Just one point on that. So are your data reported in ontology so that if we are working on a species, we would easily know that this gene in the rhino or the camel is involved in such or such adaptation mechanism? Because generally we don't go to all possible species, although you are only in mammals. Because if we want a rhino to fly, I don't know whether we could transpose from bird, but well, at least for mammals, maybe it's a question for also EBI. Where could we find all these annotations in the same place?
Yeah, so and maybe this was a question for like Luca's level of expertise, and I'm trying to understand it the best that I can. So the way that Lucas did, was excluding genes that were specifically for like a spro gene or rhino gene, and not looking into that, and doing the annotation based on the human, what is known for the human, right? So a lot of the genes that we see here that they're famous or they are well studied because they are human derived, right? So I think that's one of the topics that was commented before day or the day before about us having a better understanding on the functional annotation of species specific and not only based on humans, right? I'm not sure if I answered your question. Yeah.
Thank you. Last burning question or yes? June.
Really nice talk, definitely entertaining, and also really nice story. I have so much of the data interesting. I'm just wondering, besides that fibrous those, do you guys connect any other sample like blood from those individuals?
Not at this point. So what we're doing, at least not for those species. So what the frozen zoo has is only fibroblasts. And there is a discussion of us improving our biobanking efforts for other cell types. That's kind of like in the discussion and hopefully on the horizon. Some of the cell lines that we use for this specific experiment were established in Alison Hindle's lab. And because she had a NASA grant for studying hibernation, some of the species, she also has cell lines or primary cell lines for other tissue types. Like one of her students did with brain, muscle, and so on, trying to get differentially responses among different tissues and cell types as well in a single organism. So there is an effort to do that, but the material that I have access thus far does not involve other cell types.
Yeah, because I'm wondering, because the blood, right? It's a pretty common across all of these, and it's relatively easy to connect if the animals go alive, some of the information, particularly more epigenomic assays that will provide a reorganized,
You know, you can look at the functional information for comparison. Because
for other
we
animals.
have lots of information from the blood.
Yeah, for sure. A lot of the mammals, I would say half of the mammals that we work, the materials were coming from collaborators around US. And I think those collaborators definitely have access to other cell types, tissues, and cell lines. The ones, the frozen zoo, you know, this is it's very complicated to get access to other cell types for they don't like to do a lot of messing around with the animals as well. But
So
you know,
this animal, sorry, this animal is not from big.
well, I can show you real quick where it is. 3D pictures, so you can have a better idea where you're coming from. Okay, so the two camels are fibroblasts from San Diego Zoo. The two bats are from collaborators, so those are caught in the field, and then they send us the biopsy first to establish the cell lines. Those squirrel and seals were also established in the Hindel's lab. Rats as well. The humans we bought, the primary fibroblasts. Honey badger, dolphin, rhino from the frozen zoo, Lada from a collaborator, and humpback whale also from a different collaborator. So, you know, for those ones that come from other labs, they're collecting because they have access to the animals, so they collect as much material as they can, right?
Thank you.
Thank you very much. I think we can thank you again and wish a good evening to everyone. Thank you very much.